All right. Thank you, Dean Hu. Thank you, Provost Wendland. Thank you, Jonathan. We have a lot to discuss still. I appreciate every time I have a chance to hear Jonathan give an introduction. I will go to any talk where Jonathan wants to introduce the speaker, except my own introduction. So I really want to welcome everybody who's here in the room, and thank you all for coming. I also want to welcome all the folks who are online, especially want to give a shout out to our friends who are home who couldn't come because of COVID, because of hydrocephaly, because of foot surgery, because of depression. I want to wish them all rapid healing. And what I want to talk about today is really a celebration of the science that we do, the scientists who do the work, and those of us who have gotten involved in trying to communicate the science to the public. And I really appreciate the chance to be honored with Viktor Hamburger's name. I want to thank the family of Viktor for this remarkable legacy that Viktor gave us. And you heard already a great talk. I really could just stop here and say, you know, do you have any questions after Jonathan's introduction? But Viktor was really a remarkable person who was clearly well loved here at Washington University. When I arrived in 2000, he was 100 years old, and I didn't have a chance to meet him. But when I arrived, I was lucky enough to be here when we held a symposium in his honor. And at that symposium, I learned a lot about Viktor's research, which intersects with the research that I'm going to tell you about today. You're going to hear me talking about cells communicating with cells. You're going to hear me talking about development and synchrony. Everything for my lab is about synchrony. And the other thing I want to stress is that I appreciate that as scientists, we are part of a family tree. And here I'm showing you, if you have really good eyes, you can see part of Viktor Hamburger's family tree. These are the trainees who followed from Viktor's lab, including a Nobel laureate and many other people. And this family tree ultimately intersects with my own training. And I really appreciate that Viktor gave us the Department of Biology that we now know at Washington University, and really created a community here that celebrates people who come from diverse backgrounds and wants people to challenge the central the dogma in the field. So I'm going to take a couple of minutes now to just talk about the research that we do in our lab, and I'm going to need you to participate in a small quiz. So how many of you went to sleep last night? Raise your hands, please. Almost everybody. Fantastic. How many of you woke up this morning? All right, we've got a good consensus. Almost everybody's awake now. How many of you plan to go to sleep tonight? Almost everybody. And tomorrow, how many of you hope to wake up tomorrow? A very optimistic crowd here. And what I think you all realize is that you are living a rhythmic lifestyle, that this synchronization to life on Earth, synchronizing to our local light-dark schedule, makes us a different person in the morning than we are in the evening. And that's what's fascinated my lab. So next question in the quiz, when did you wake up on Sunday? Your last, let's say, free day. How many of you woke up before 8 a.m.? Raise your hands, please. Oh, it's an interesting demographic. Look around the room. Keep your hands up for just a second there. Look around. Just get an estimate of who your friends are at 8 a.m. Okay, hands down. How many of you woke up between 8 and 9? Hands up high. Be proud of yourselves. Remember, science only proceeds with honesty. Okay, 9 to 10, any hands up? Okay, yes, be proud of yourselves, okay? 10 to 11, anybody waking up? Has everybody woken up now on Sunday? Yeah, Jacob, please, what time did you wake up? 11:15. Thank you for that. And I'll just have you note the young person in the crowd is preferring to wake up later in the day, okay? And so the next question is, what woke you up this morning? So can anybody just shout out what woke you up this morning? Dean Wendland? Preaching the gospel. Provost Wendland says she woke up just her own circadian rhythm. How many of you used an alarm clock this morning? Raise your hands high. Look around. You are all waking up unnaturally. And so the rest of this talk is about, what is it that would wake us up if we didn't have an alarm clock? We didn't have our moms to tell us it's time to go to school. And we can illustrate that you do have a built-in biological clock that is there to help you schedule your day by doing something like this. You take a mouse in a cage with a running wheel, and you just monitor when the mouse is awake by watching it run on the wheel. And our record holder here in our building ran about 13 miles a day on a wheel. And that mouse, our mice tend to want to run in the dark. Here you see the peak of activity in the dark. And then they rest predominantly during the day. If you do this same sort of experiment with a human, like watching their Fitbit activity, and you take away all of their environmental cues, you put them in a cave, you take away their alarm clock, you take away the clocks, and you ask them, just live your life, they will still wake and sleep on a roughly daily schedule. But if you're a typical wild type human, you will wake up every day about 15 minutes later than you did the previous day. So if you wake today at 8 a.m., tomorrow will be 8:15, the next day, 8:30. And that's because you have an internal circadian clock, a clock that has a near 24-hour period of about 24.25 hours. If you're a mouse in our laboratory, a typical wild type mouse, it's 23.7 hours, plus or minus about 4 minutes out of every 1,440 minutes per day. It's a remarkably reliable, precise timing mechanism, which has made itself amenable to asking questions about how these rhythms arise. And so here we have evidence in constant conditions in the absence of the Earth's timing cues like sunrise and sunset of an internal body clock, a circadian clock. And much work in the field has resulted in us understanding that this is a genetic program, something that you inherit from your parents, and you can blame your sleep schedule on your parents, in part. And so what we know now is that of your 20,000 or so genes in your genome, about a dozen of them are dedicated to telling us when to wake up and when to go to sleep. And illustrated for you here is a single cell with one of those clock genes, a gene called period, which is expressed at certain times of day, and then its protein accumulates and goes back into the nucleus to turn off its own expression. So the message goes away, the protein goes away, the repression goes away, and the cycle can start anew, a near 24-hour cycle. If you want to learn more about this, you can take my class. It's called Biological Clocks. It's offered on Tuesdays and Thursdays next semester. Alternatively, you can go to Wikipedia. So I want to tell you a quick story about how the science that we do here at Washington University has been really something that I've been able to use in the classroom, and something that Viktor Hamburger really celebrated, the idea that research and education should go together. And one of the things we do in this class is we have students vote for and select topics to put onto Wikipedia, things like scientists, genes, and key concepts. Some of you in this audience have been the subject of Wikipedia pages generated by students in this class. We're very proud of this project, and in 2017, Wikipedia wrote an article about the students in this class, when 3 of the previous Wikipedia sites that we generated for 3 people who ended up winning the Nobel Prize were noted for sudden bursts in hits. So these 3 very famous scientists have been going along at about 10 hits per day on their Wikipedia sites. And then on the day the Nobel Prize was announced in medicine or physiology, they went up to 25,000 hits in a single day. And then over the next few days, they learned just how long fame really lasts. So I was really excited about this, and I talked about this with colleagues, and my field has really been helpful in supporting the students in this class in continuing to keep information flowing from what we discover in the field to the general public. And it caught the attention of a young scientist in Israel, a woman named Rona Avram, who has decided to make this her new field. She has generated a new field of history of science, based upon how information is being added to Wikipedia, with the premise that as scientists debate concepts, they are put into Wikipedia and removed from Wikipedia until they are considered to be the consensus opinion. And so here I'm plotting for you data that Rona generated just last week using a new tool that she has developed that combs through Wikipedia and tells you how many new Wikipedia sites were generated each year in a field, and you can pick your topic of choice. Here she chose the word circadian. What I hope you can see is that we started our work in 2000, and over the years, we've been adding information to Wikipedia. And there's a really interesting biannual cycle that's now started to establish itself. Those are the years that my class meets, and we have a Wikipedia project. And so as a field, we have gone back and we are continuing to edit what is out there, and students at Washington University have become instrumental in helping us continue to convey the current state of knowledge to the general public. I'm going to show you two data slides now from research in the lab. The lab has addressed many different questions. One of the questions is, where in the body do these rhythms start? Is it the case that individual cells can really act as circadian pacemakers? And this is an experiment that was done by a graduate student in the lab, Alexis Webb, and an undergraduate in the lab, Nikhil Angelo, who I was really pleased to see made it here today. In this experiment, they're monitoring gene expression on the y-axis, and we measure that over time, and you see this cell turns on and turns off, turns on and turns off, turns on. And at this point here, the experimenters went in and they poked all the cells in the dish except for one. They killed them all, except one cell. And that single fully isolated cell was able to keep on ticking, expressing circadian rhythms. So this clockwork is intrinsic to individual cells. The machinery is truly intracellular. We've gone ahead and continued these sorts of experiments with different cell types in the body. And we found that this is true for different kinds of neurons in the brain, different kinds of glial cells in the brain. And we've even gone outside the brain and we've started looking at things like cells within the liver, cells within the uterus, cells within the cervix. And this has led to collaborations with people like Sarah England here at Washington University and OBGYN, where we've begun to ask questions about, when do clocks develop? And how do these clocks in the mom and in the fetus contribute to important things like the timing of birth, as you heard from Jonathan? So this, additional cell type that I want to mention is chondrocytes, a new collaboration with folks in Farsh Guilac's lab here at Washington University down in the medical school, looking at whether we can understand how circadian timing can be used to treat things like arthritis. And so the next question we want to ask is, okay, if individual cells can generate rhythms, how do they communicate that timing to each other so that you are a coordinated clock shop and all the cells are synchronized to each other? And work in the lab has focused on the role of secreted factors from cells called neuropeptides. And here I highlight two, a green one that I'm going to focus on here, easy to remember, VIP, very important protein, okay? Or vasoactive intestinal polypeptide for the aficionados. VIP is expressed in a tiny population of cells that are located in the base of your hypothalamus, right about here, about 1,500 cells on the left side of your brain, 1,500 cells on the right side of the brain. And when those cells are there, the cells in their neighborhood are perfectly synchronized, all beating, expressing their daily rhythm and gene expression for as long as we can record. Literally a year in a dish, these cells will stay synchronized to each other. If we knock out VIP, the cells can still express daily rhythms, but they can no longer synchronize to each other. They lose the ability to talk to each other and agree on local time. So we illustrate that here with the schematic, where there's an intrinsic clock within each cell. That cell secretes an important factor that tells the other cells around it what time of day it is. So if the cell on the left says it's noon, the cell on the right might say, oh, I thought it was 11:50, I'll adjust. And so the cells will all synchronize and agree. And finally, I just want to mention that this work has taken me and my lab in surprising directions. We found ourselves now working on the clinical relevance of biological clocks. And so I want to just highlight one project that has been really influential for me. We've focused on glioblastoma. In the last 10 years, a collaboration that started with Josh Rubin here at Washington University and a clinical oncologist, John Campion. We found that glioblastoma, brain cancers, have intrinsic daily rhythms, just like other cells. And we found that these rhythms impart a change of sensitivity to the drug of choice, so that the same tumor is actually responding and dying to the drug of choice in the morning and relatively insensitive to that drug if you give the drug in the evening. This is called chronochemotherapy or circadian medicine. And here at Washington University, we're running the only chronotherapy clinical trial where we are testing temozolomide in the morning versus the evening on people with this devastating disease. So I just want to conclude now by thanking the people that really make this work possible. Of course, all of these data were not generated by me. These were generated by the engine in the lab, the people who've been a part of the Herzog Nation. I want to start by highlighting two people who've been really dear to me, Daniel Granados Fuentes, here, and Tatiana Simon. They've been with me pretty much since we started the lab. And they've been just, I honestly could not do what we do without the two of them. In addition, we've had 12 postdoctoral fellows, 12 graduate students who have all gone on to careers, our technicians, 68 undergraduates, and many high school students who have contributed to the research that we do and have really made me realize I'm naive and my hypotheses are a great place to start, but these people who generate the data are really the ones who are making the discoveries. I also want to thank my colleagues here at Washington University and in St. Louis. When I arrived in 2000, we had a group of researchers, myself, Paul Taggart, Russ Van Gelder, and Lou Muglia, who formed a small journal club. And our labs would get together every week to talk about new data in our labs and new publications in the field. That group has now grown to something like 30 different labs. We have a vibrant multi-university meeting, which we call Clocks Club. And this group studies everything about this clock system, from the pacemaking cells and how they synchronize to each other, to how they synchronize to the environment, and how these regulate just about every hormone in your body and every aspect of how you feel in the morning versus the evening. I also want to give a shout out to my support network. I really couldn't be here today without the people who have been my mentors and my support team. So I came to Washington University in 2000 under the guidance of a guy named Bill Schwartz, who told me, you want to go to Washington University, where I had choices. He said, you will be happy and thrive. And he was right. I could not have imagined a more welcoming, supportive environment. I also want to thank Gene Block, who is the chancellor at UCLA. But when I was a postdoctoral fellow, he was my mentor in the lab and really rescued me as a scientist and taught me how to be a scientist that asks questions that are exciting and tests real hypotheses. I am rescued on a daily basis by the diligent efforts of Jan, Linda, Andrew, Melissa, and Jen, who make sure that our research is done right, our grants are funded, the money goes to the right places. When I arrived at Washington University, I formed two close friendships. Doug Chalker and Jeff Zox came to the university at about the same time. We ride bikes almost every Saturday together. And if you hear three people biking by talking about science and seeming to ignore traffic laws, it could be us. I apologize. I want to thank the three chairs of biology, Ralph, Kathy, and Joe, who have made biology a really wonderful place to be. I want to thank Barbara Schall for nominating me for this honor, Feng Sheng and Beverly for making Washington University a very exciting place to be right now. And then I finally want to thank my family. I want to thank my parents who are here. Thank you. My biggest fans, and I am their biggest fan. I'm really lucky that my sister Carolyn is here today. She puts me in my place. And if you could please give it up for my wife, who has given up a lot for me, Mary Barjon. Among the many things I have learned from my wife, it is that sleeping in is a reward. And I want to give a shout out to my two boys, Max and Isaac, who make me very proud every day. I started this talk by talking about how we're different people in the morning than we are in the evening. Our mood changes from morning to evening. But for some people, that's not the case. They are depressed all day long. And that was the case for my sister, my little sister, Elise. And in the words of my sister, Carolyn, we need to do better as researchers and members of society in recognizing depression, mania, and bipolar disorder for what it is, a disease. And so with my sister, I'm really pleased to share with you that we have started a new fund, which we call the Erik Herzog Mental Health Research Award, which we will implement through the Department of Psychological and Brain Sciences, PBS, under the able leadership of Jeff Zox here, which will recognize young researchers moving to recognize, treat, and improve the outcome for people living with mood disorders. This talk is dedicated to Liesl. Finally, I want to give a shout out to the first Viktor Hamburger Distinguished Professor, Sally Elgin. And Sally, if you're out there, I just want to say thank you for being a role model. When I arrived at Washington University and she was bestowed with this honor, I remember her saying that it was a perfect honor for what she wanted to do next. Sally was dedicated to finding ways to use research to teach students. So learning through research experience. And I have been bitten by that bug here at Washington University. And so it's really a great honor to be the next Viktor Hamburger Distinguished Professor. With the help of people like Rochelle Smith, Diana Jose Edwards, and Lori Corzine, as you heard, we started and run a program that's now got over 60 undergraduate alumni who have gone on to PhDs or MD PhDs in neuroscience-related fields, a program we call Endure. With the help of Vicki May and her Institute for School Partnerships, we have run programs like the St. Louis Area Brain Bee. You guys are all invited this February to our 13th annual Brain Bee. We have over 100 high school students competing from over 40 different high schools in St. Louis. We send the winner to the national competition. A few years ago, our local winner won the national competition and went to compete and represent the United States in Berlin. And finally, thank you. And finally, I just want to thank Annalise Schaefer and the Office of Neuroscience Research for unifying the neuroscience outreach efforts at the university and in our community. As you will see, there is a tremendous neuroscience community here at Washington University. And through our outreach activities, we contact over 4,000 people a year through the Science Center and contacts in schools. If you want to learn more, you can go to neuroscienceoutreach.wistal.edu. And so I will conclude now with an explanation of my title for this talk. It was not a misspelling. It was just a jumbled title on purpose. This was a derivative from the Ernest Hemingway novel, For Whom the Bell Tolls. And that was intentional. And I went back to the book to look for a quote for you all. And I was surprised to learn that I had not memorized this quote. In the book, he says, "But in the meantime, all the life you have or ever will have is today. Tonight, tomorrow, today, tonight, tomorrow, over and over again, I hope." And so every day that you hear WashU's chimes ring here on campus, I want you to think about what can I do next and enjoy the privilege that we have here at Washington University. And with that, I will get out of your way and we can have some snacks. Thank you.